Oof, recording.
So advanced techniques, which confocal microscopy is part of. So, what is confocal microscopy? It's okay? Yeah, you want to take it? It's okay. Okay, confocal is just, it uses an aperture. You're gonna be the funny guy in the video that drops his thing all the time. Okay, that's good. So with the confocal, you use a little aperture, and it's called a pinhole, which is a pinhole. Really small things that allow light, which is only the light that occurs at the focal position. So let's say you take those rays here would be focused and they will be blocked by this small aperture. Same thing if you take a ray that are on top of it, they will be blocked also by this aperture. So only rays which are really at the focal position can enter the pinhole and be detected by the detector. And so with confocal microscope, you have, in one case, you have only one pinhole, which is, most of the time you use a laser, but it's scanning. So you have laser scanning microscope. So many of them are called LSM, or laser scanning microscope. So you have those mirrors which will oscillate and make a small point that go onto your specimen and scans it. While you have the confocal spinning disc, so it's a disc which has many pinholes like this. So you have small dots, and then you have a light source that goes onto your specimen, and this you have micro lenses which will focus light into that little pinhole, go onto your specimen, come back to that pinhole, and be splitted by the dichroic and go to your detector. Okay, and this spins, so it turns and make some lines onto your specimen, and those lines are configured in such a way that all your specimen is illuminated proportionally, so we'll have the same amount of photon that arise onto your specimen, and so you see. So this principle is called the spinning disk confocal. And with confocal microscope, so because you only take the light which occurs at the focus, you can move your specimen into that focus position and take 3D structure from it. Okay? So with a confocal microscope, you can cut your specimen into light planes and take all those light planes and make a 3D from it. And I think this is supposed to be a video. Not sure. We'll see. Where's the mouse? Look, oh. It's not there anymore. Oh, here it is. Okay. Plane is there. Yeah. So you have a 3D structure. It's only a small little cell with surface marker that you see there. Okay. Confocal microscopy and what is called multi-photon microscopy. This is a really nice technique. where you use the fact that if you throw a bunch of photons, and when I say a bunch of photons is many, I don't know the numbers, how do you call it when it's big like that? 10 with maybe 15, 20 zeros, photons, okay, so a couple. And when you throw that amount of photon at the same time, and they arrive at the molecule exactly at the same time, the molecule can not only absorb one photon, but it absorbs, let's say, 2 or 3 photons. That has in the infrared range energy. And since it absorbs 2 or 3 photons, the energy that it absorbs will be addition. It's going to be an addition. So you will add the energy of each photon, and it's the equivalent of, let's say, a UV photon. So you take infrared, you shine it onto your specimen. Your specimen will absorb not only 1 photon at a time, but it will absorb either 2 or 3 photons, okay, or maybe 4 or 5, but it's going to be really rare if you absorb that much photon, because the more photon you absorb, the more photon you must shoot at the same time and be the exact same time and the same absorption. Okay, so but this probability is really rare, and it happens when there is a lot of photon. And where there is a lot of photon at the same time, it's really at the focal position, because every photons are sent in a certain focal position, and at the focus, this is where all the photons meet and are in a particular place in space. Okay? And so the event of absorbing 2 or 3 photons, the probability is only happening at the focus of the lens. So if it does absorb photons, it's going to be at this exact position. So this thing here is shooting infrared pulsed bunch of photons, and here you see the absorption of only photons at the focal, while this guy here is shooting photons that are absorbed all the way along into that cuvette that has a fluorescent marker. So here it's really 1 photon absorption, and the 1 photon absorption occurs everywhere. And you see that it occurs rarely in the back here, because the absorption is mostly done at the beginning, and then there is many absorbed photons, and here there is no more photons, so there is no more absorption. So you only see light from here. But here, photons are not absorbed, and only at the focus they are absorbed. Okay? So confocal microscopy using, actually you need pulsed laser, because you need to shoot so many photons that if you have a continuous laser, so a laser that shoots photon at the same time that can produce multi-photon absorption, you're going to kill everyone with it. It's too many power if it's not pulsed laser. Even pulsed laser is, I think the pulse is in the terawatt. So you have many millions of watt that occurs there at the precise time. Okay? So it's only with pulse laser that you can achieve that. And so here we don't have any pulse laser in the institute, so we don't do any multi-photon microscopy. The laser by itself worth a couple of $100,000. So it's about $250,000, I would say. just the laser source by itself. So, but it's a nice technique, and it is nice also because you use infrared that you shoot onto your specimen, and infrared are not absorbed by a living specimen, except at the focus, where you get that probability to have 2 or 3 photon absorption. Optical projection tomography. So this is another technique where you use, well, you know about tomography mainly with X-ray, but you can also do it with light, regular light. So the basic principle of it is that you have a specimen that you make turn, or you turn around the specimen. But the basic thing is that you look at a specimen with many different angles. And from there, you can figure out how it looks in 3D. And this is basically the technique. Maybe you've heard about deconvolution. And maybe in maths, you've heard about convolution. Maybe not. So it's a nice term, and it's just a mathematical thing. So a convolution, it's a product of convolution. Usually it's 2 operations. So it's a multiplication of 2 terms inside an integral. So basically that's a convolution. So it's a product of something into a summation of things. And in image, when you produce an image, you always convolute something in order to see the thing. So let's say, well, with the halo here, halo, which is the French part of hello. So in English, it's H-E-L-L-O. In French, it's halo, A-L-L-O. Simpler. So let's say you have halo like this, and the halo would be your object. So every pixel which composes the letter A here is either white or black. Okay? And let's say you have a function that for every white pixel, you see this pattern. Okay? So you have a white pixel here will produce that pattern. Another here will produce that pattern, and so on. So the final image that you're going to see through something that produces this from 1 pixel, you're going to put this function over it and do a multiplication of it. It gives this, and you're going to add all those functions, and you turn out with an image like this, which has kind of a shadow surrounding it, because for every pixel it produces a ring of shadow like this. Okay? So that is the convolution. So it's a multiplication of this function for every pixel here that produces the final thing, which is the product of convolution. Okay? And when you look into a microscope, every dot of light produces a pattern like this. So if you have a probe which emits light, it will produce a pattern like this. So if you have many probes everywhere, every probe produces a pattern like this. It's going to end up like this. So when you look at the microscope, it's not as obvious as this, because it looks like this. And here you don't see the rings almost, because you have a big spot here, and already here the image is kind of intensified to see the rings. Okay? But when you look in a microscope, you're going to see things like that. So this is the image that you acquire, but the real object is this. Okay? But what you see is this. So when you get an image, you see this. And you know that the real object is this. So because you know that each dot of light produces this, then you figure out, oh, maybe I can get this from this. And this is the deconvolution. So you go back to your original object, and this is what you try to get. So this technique is called deconvolution. So it's a mathematical process to get back to the original object from the image, knowing this thing here, which is called, you can note it, maybe I didn't write it anywhere. It's the PSF, which is the point spread function. So it's this function here, the point, we call it point spread because it's the point that spreads out, and it's the function that shows it. So the point spread function. Super resolution techniques. And so, nowadays, so you're limited by this point spread function because every dot of light that you look at is big like that. So if you have one dot which sit right across the other, you cannot distinguish it because every dot produces a big dot like this. So if I use my marker and I put a dot and I put another dot right across the other one, you cannot really know where the center of each dot is because you have kind of two mixed things. Okay? And so super resolution techniques kind of arise and it's since maybe 1993, I would say, or 1994, that some people, they did have the idea of looking at their specimen into different time point, or kind of. And there's this STED microscopy, which you can scan your specimen and even use and get more details. And it's a tricky way to get more details that I will explain a bit later in the slide. So those guys, so Stephen Hell, Gustafsson is dead now. Pretty recently, I would say. I think it's near 2000 or something like that. And there is also, what's his name? The other guy. There's a third one, which is mostly for Paul Storm D. Storm, but I didn't put the right name. But those three people, they won the Nobel Prize, I think, in 2014 for their techniques that are now widely used in microscopy. We don't have any super resolution microscope here yet. I guess we will get one at some point. So the first technique is called structured illumination microscopy, SIM. And this technique uses a different lightning pattern onto your specimen in order to get more information, and it's all a mathematical process of many images that are acquired in order to get better resolution by a factor of 2. So let's say you have 1 pixel in regular microscopy. With SIM, you're going to get 4 pixels instead of 1 for the same object. Okay? So you get a factor of 2 in both directions as well. STED microscopy. STED uses two lasers. So one laser is used to excite the probes, and the other one is used to what we call deplete the probes. So once you excite, you can also shine a laser onto some probes in order to bring them back to a ground state. And this is basically what it uses as technique. So it excites it at a certain point in space, and then it uses kind of a donuts, which will deplete the probes all surrounding the focus, and it ends up with only few particles, smaller region that can produce your fluorescence, and this is how you get better resolution. The other techniques is palm storm. It's a stochastic technique. So let's say if you image all molecules at the same time, you have something blurry like this. But if you have a way to make them excitable not at the same time, then you excite a couple of them which are apart each other. You find out their center, and then from all those centers you produce a map which you have only the centers that will make something much more clear. Okay? And so super resolution techniques, they use that fact that you don't look at or excite probes at the same time. Same thing with the stem. You excite it, and right away after you de-excite a couple of them, and you just look at the one which stays excited. Okay? This is how it works. So that is mostly super resolution techniques. Other source. So this is basically where you can find a lot of stuff. Microscopy U is a nice website where you can find out many references. Okay, so now I'm just gonna show you with pictures what the core facility looks like. So what kind of instrument that we do have. So again, the spinning disc and confocal. So again, you have that draw that show you that only the focus will go through the pinhole here and be detected by the photomultiplier tube. So we have the LSM 700, LSM 710, which are laser scanning microscope, and we do have the spinning disc confocal that does have the Yokogawa spinning disc. This is the spinning disc technology. So we have 3 confocal. There is another one which is not shown here, which is just the room beside here. And we'll show it to you maybe after the course if you want. So it's another laser scanning, which is another brand. So those one are Zeiss microscope. The other one is a Leica microscope. Basically same principle. Local. So room 1425, which is just in front of this room, has many other microscopes. So a regular dissecting microscope. So we call it dissecting microscope because you have enough room to produce your or dissect. Okay, and usually it's binocular. It gives you a kind of 3D view of your specimen. This is just a regular bright field microscope. This is an optical tomograph. So you can put your specimen here, and there's a motor which will make it turn, and the optical system is still here, so you can produce 3D also from this. This is just an image processing workstation. We do have a laser dissecting microscope. So it has a laser device that you can control the position of. So you can have a specimen and you draw on the screen where you want the laser to cut your specimen, and so you can get a precise portion of your specimen under the microscope from the laser dissection that you do. Actually, this one is now at the 5th floor. Another room where you do have just regular fluorescent microscope. This one is inverted, this one upright. There's also the high content screening. So this is a device where you can just put your Petri plate. Usually it's a 96 well plate. So you put your specimen into it. You can have many plates, so it has a robot arm that take the plate, put it in the system, and take many pictures into many wells. So everything is motorized, the stage is motorized, and so it can take many different images from many wells, and this is why it's called high content screening, because you acquire many data. And there's a software also that will analyze or count cells or granules into the system. This is just a regular microscope in the same room. And look out, so room 5750, where you have many other microscope mainly used for fluorescence, and two inverted microscope where you can produce time-lapse movie. So time-lapse is just you take many images at a different interval of time, and therefore you can produce movie. This is just an example of a scratch essay. So time one, you just scratch your culture cell, so you produce a scratch, and with time, cells gonna close that scratch, and you can figure out how fast it goes, in which direction cells are moving, and things like that. Again, fluorescence. So this is just a Jablonski diagram showing energy levels about fluorescence, and also images you can produce. This is a retina. Many nucleus here, ganglion cell, I think. I'm not really sure about that. Yeah. This is just an example of image of Nomarsky. So you can produce Nomarsky at the confocal microscope also, because it has laser source which are already polarized. And you can use the transmitted light polarized. So we do have transmitted PMTs that allows to produce that kind of contrast with all the confocal that we do have. Dark field. So maybe, is it the video? This is the video. Yeah, so you remember I explained where is the video? No, it's not. Well, it was. Sorry about that. I'm pretty sure I produced the video, but doesn't want to play it up here. Okay, yeah, I will continue. This is just really, so maybe I have it somewhere. No, this is, play the video, please. Please. No. Okay, well, I could find out the videos after. Yeah. So and this is kind of the software that we do have. So there is Imaris, which is really, really nice for 3D rendering. Velocity, which is mainly used for acquisition here, but you can also use it for annotation. Norton Eclipse, which is rarely used now. It was a nice software maybe 10 years ago. Now it's not so useful. But molecular device, that's basically what you use with the high content screening. And MATLAB use MathWorks, which is the producer of MATLAB, which you can interact with Imaris. So you can program, if you program, or I can program something for you in MATLAB that you can use. And I can show you also how to use MATLAB to program. It's really nice. And maybe this is the video, no? Again? No. One more. That's 3D that you can produce with Imaris. And so the service that I do offer for you is mainly training, just advice, and we can find out a solution to analyze your, process your image, and produce user interface also to make your task easier. That's okay, the confocal panel that we already talked about. Yeah, maybe just something that picturize a bit better maybe. So you have an X scanner and a Y scanner, so those things turns and make the way to vibration. Okay. So you can scan your specimen. And the convolution again, yeah. So when you get a smaller point spread function, while you can have really bigger point spread and a bit smaller point spread. So this would be, this would be low numerical aperture, high numerical aperture, and middle numerical aperture. If you see the blurring that occurs depending on how wide is your point spread function. And yeah, if you have questions, yeah, we're pretty good. So you have 15 minutes for you. Well, I think you will get maybe many questions during the, I think it's 3 weeks for microscopy. There is this Thursday and Friday where there's no course at all. So tomorrow I think we get another course, mainly to produce your account, and it's going to be pretty easy. So you can figure out that it's not going to be 2 hours, so you can plan other things. Yeah, but at least be there, I think about 9:30 tomorrow. So tomorrow 9:30 we can meet at the room just there, 1425. I don't know if it's written in the papers. Maybe. Yeah, I will send an email if it's not the right room. But it's going to be close to my office. Yeah. Or 15750, I don't know. I will go where it's written that it is. So that's it. That's good? Yeah. Okay.
Should we really focus on the working of all those microscopes?
If what?
Like you showed advanced techniques and all.
Yeah.
So like, should we go back and focus more on how they work, or?
No. There's not going to be any exam about that. The important thing is just to, I think, to listen at this. That is first thing, and I think you listen pretty well. You're only 3, and I can catch you if you're not listening. But we will go on the instrument, and this is where maybe you're going to have more question, and I'm going to try to point out what I just tell you here. Make sure you understand. You will not understand everything right away. It's normal, but at least that you get that numerical aperture is an important thing. The way you light your specimen is important. Fluorescence, those things. Yeah, but yeah, there's no big stress. It's gonna be fun. It's important to have fun. Yeah. Good. Excellent. So see you tomorrow. And if you have any questions after, so I will send to. Is it Virginie that communicates with you? Yeah. I will send her the PDF, or maybe you already received it. No? Okay. So I will make sure that I send the PDF of this talk that I just gave, and yeah, that's it. And maybe the video if you're interested to listen to it again or sleep well. Okay. Good.
What's really going to happen on Thursday and Friday? What is IRCM retreat?
Oh yeah, ISCM retreat. You're not part of it, huh? I think. Or yeah, it's actually usually the years before it was only the PI, the principal investigator, which would take part into that event. But last year they decided that they would make students, some of them participate to that event. So, and we go somewhere else than here. I don't remember exactly where. But just to talk about the research and make sure that every group know a little bit about the other one and increase collaboration into the institute. Yeah, so that's basically what it is. Good. That's it. Yeah,